order. Hearing is now resumed.
Good morning, Mr. Commissioner. It's Patrick McGowan. With me is Micah Carmody, counsel for the Commission. Today's hearings will focus on the topic of municipal wastewater. We have three panelists with us this morning, starting on my left, Dr. Ken Ashley, moving to the right in the center seat, Dr. Peter Ross, and on the far right, Graham Van Agellen. Mr. Ashley is not affiliated with any government department presently. Dr. Ross is with the Department Fisheries and Oceans, and Mr. Van Agellen is with Environment Canada. Perhaps we could have the panelists sworn.
Solemnly affirm that the evidence to be given by you to this hearing shall be the truth, the whole truth, and nothing but the truth?
I do.
I do.
I do.
Would you state your name, please?
Ken Ashley.
Peter Ross.
Graham Van Agellen.
Thank you. Counsel?
Thank you, Mr. Commissioner. I'm going to seek to have each of these witnesses qualified as experts, and I'm going to start with Dr. Ross. I'm going to seek to have Dr. Ross qualified as an expert in aquatic toxicology. Dr. Ross, you have a PhD in ecotoxicology, which you attained in 1995.
That's correct.
You also have a master's degree and a bachelor's degree in biology.
Correct.
Your present position is with the Department of Fisheries and Oceans, where you are an environmental toxicologist based at the Institute of Ocean Sciences.
That's correct.
You've been employed by DFO since 1999.
Correct.
And your area of research interest is ecotoxicology and the risks to aquatic life associated with certain chemicals.
That's correct.
And on the screen we have Dr. Ross's CV. I'm going to suggest that be the next exhibit.
Exhibit 1043.
Mr. Commissioner, based on that examination, I'm going to seek to have this witness qualified as an expert in aquatic toxicology, subject to any other questions that might be posed.
Thank you very much.
Next, Mr. Van Agellen, I'm going to seek to have him qualified as an expert in toxicology and toxicogenomics. Sir, you're presently the head of the environmental toxicology section in the Pacific Environmental Sciences Center of Environment Canada.
That's correct.
You've held that position since 2000?
Yes, sir.
And you've been with Environment Canada since 1996?
That's correct.
Prior to that, you worked with the BC Ministry of the Environment for a number of years?
That's correct.
You hold a Bachelor of Science in Zoology and Biological Sciences?
That's correct.
And through your experience, you've developed expertise in the testing of effluents for toxicity to marine and freshwater species?
That's correct.
And I wonder if you could just very briefly tell the Commissioner what toxicogenomics is.
Toxicogenomics is the study of the gene expression within within aquatic organisms. Specifically, I work with trout and other salmonids. We look at the gene structure, the molecular structure, to look at potential effects as a result of ultra low level contaminants.
And if we could have Mr. Van Agelen's CV marked as the next exhibit.
Exhibit number 1044.
Mr. Commissioner, based on that examination, I'd seek to have him qualified as an expert in toxicology and toxicogenomics.
Yes, thank you very much.
Thank you. And finally, on the qualifications, Mr. Commissioner, we have Dr. Ashley, who I'm seeking to qualify as an expert in three areas: environmental engineering, aquatic ecology, and limnology. Now, Dr. Ashley, I think the Commissioner has heard a fair bit about environmental engineering and aquatic ecology, but I'm not sure the term limnology has come up yet. Could you just briefly explain to the Commissioner what that is?
Limnology is the scientific study of inland waters, freshwater oceanography, for lack of a better term.
Okay, thank you. You have a PhD in environmental engineering?
Correct.
You have two master's degrees, one in environmental engineering and one in aquatic ecology?
Correct.
You're presently an instructor at BCIT in the Ecological Restoration Program?
Part-time, yes.
You're an adjunct professor at UBC in the Civil Engineering Department?
Correct.
And you also work for Northwest Hydraulic Consultants as a senior scientist?
Correct.
And you've previously worked for both the GVRD and the province of British Columbia?
Correct.
Okay. And is that Dr. Ashley's CV on the screen?
It is, and there's personal information on the top of the page.
Fair enough.
Scroll it down.
Thank you. I'm going to suggest that this exhibit be the next, this CV be the next exhibit, Mr. Commissioner, and perhaps, with your leave, we could, at a convenient time, replace the exhibit with one with the personal information blocked out.
That'll be Exhibit 1045.
Thank you. Mr. Van Hegelen, I'm going to start with some questions to you about the Canadian Pacific Environmental Sciences Centre. Could you just briefly explain to the Commissioner what that is and where it's situated within Environment Canada?
The Pacific Environmental Science Centre is the Pacific and Yukon region's key laboratory for toxicological and analytical chemistry analysis. It forms what I would call the Pacific anchor for Environment Canada's network of regulatory laboratories. There's approximately 55 scientists and technicians that work at the centre in North Vancouver. The lab is divided up into three principal components. There's a toxicology group, a chemistry group, which is subdivided into organic and inorganic chemistry, and there's a water quality monitoring group that's responsible for shellfish water quality monitoring sites.
And you're the head of the toxicology section, correct?
That's correct.
And could you just briefly explain to the Commissioner the type of testing conducted by your unit?
The principal function of my laboratory is to test effluents and materials that are federally responsible discharges, particularly those that are with respect to Fisheries Act 36.3 provisions, and essentially end-of-pipe discharge into waters that are federally controlled, or to waters frequented by salmon or transboundary waters.
Okay. Now I understand that to the extent there's a genomics program within Environment Canada, it's sort of run off the side of your desk within the toxicology program. Is that fair?
That's correct. Genomics, particularly toxicogenomics or aquatic toxicogenomics, is an emerging field in environmental toxicology, and it shows great promise for furthering our understanding of how chemicals or complex mixtures affect aquatic organisms. And in particular, my area of interest is how they affect fish, and we look specifically at key tissues within the fish after they've been exposed to certain contaminants or complex mixtures.
Okay. What percentage of the testing that you do in your facility is end-of-pipe testing, sort of traditional regulatory testing, as opposed to testing to support your genomics work?
I would say most probably 90% is end-of-pipe discharge.
Okay. And now, the end-of-pipe testing focuses on acute lethality, correct?
That's correct. It's the Environment Canada has a national accredited test for measuring the 96-hour rainbow trout test for measuring acute lethality to effluents.
And one of the advantages of genomics is its potential to allow you to assess sublethal impacts.
Yeah, those,
correct?
Where genomics comes into play is that the endpoints associated with a molecular expression, you can look at those effluents that are not acutely toxic, that don't kill the fish outright, but there may be other levels or other concentrations of chemicals or contaminants in that mixture that may be having effects at the molecular structure.
Thank you. Dr. Ross, you're at the Department of Fisheries and Oceans Institute of Ocean Sciences, correct?
That's correct.
And you have a particular expertise, and much of your research is focused on marine mammal toxicology, correct?
That's correct.
And while you do have experience doing fish toxicology, it's not your primary area, is that correct?
I would say that's fair. We do have a very productive collaboration with my good colleague, Dr. Chris Kennedy at Simon Fraser University, and we've been looking at the effects of currently used pesticides on salmon health with a number of different research directions there, and we've published about 9 articles on that subject matter.
Okay. Does the department's Pacific region have a toxicologist whose research regularly focuses on fish and whose area of expertise that is primarily?
No.
Is there such an expert in another region in the department in Canada?
We do have a scattering of toxicologists across the country, one in Winnipeg, one or two in the St. Lawrence and Quebec region, one in Nova Scotia, and one in Newfoundland.
Does the absence of a dedicated fish toxicologist, does that lead to a gap in either expertise or attention that's paid to this matter such as this in the Pacific region?
Well, I think it's certainly hampering our efforts to understand whether contaminants present a risk to what's happening with sockeye salmon at present. Not having that expertise means that we really don't have the opportunity to conduct research to explore the matter in depth.
Okay. In the Pacific region presently, is the department involved in monitoring or researching to determine the potential impacts of municipal wastewater on aquatic life generally, or more specifically on fish or salmon?
No, I think in general terms, following up on my colleague, Mr. Van Aglen's comments about division of responsibilities, we at the Department of Fisheries and Oceans have largely viewed Section 36 things as the purview of Environment Canada. And there has been an interest in the effects of contaminants on the health of biota within DFO, but that's largely related to cumulative impacts or non-point source impacts.
Okay. Is there anyone at the department who's tasked with assessing the actual impact of contaminants in municipal wastewater on Fraser salmon?
No.
Or Fraser sockeye?
No.
Okay. Is there such a person at Environment Canada, Mr. Van Aglen?
Specifically, there are scientists within the National Research Center in Burlington that are part of doing research, but specifically on the coast in Pacific Yukon region, there's not a single individual that solely is responsible for sockeye.
Is there a group who is tasked with assessing the impact of contaminants in municipal wastewater on the salmon in the Pacific region?
Not specifically.
Now, we heard a little bit about, a moment ago, about the genomics program that you're spending a small portion of your time dealing with. Can you tell the Commissioner how that's funded?
The genomics program was largely funded by a Environment Canada funded program. The acronym is STAGE, Strategic Technologies for Advancement of Genomics in the Environment. And that's a competitive process within Environment Canada where each fiscal year a submission into to apply for funds to continue on with genomics related work in my lab has to be done. I've been successful over the last 7 years in getting funds to build up capacity and expertise within the lab, and also have been able to partner with and collaborate with individuals at the University of Victoria and Waterloo, and also in advancing our techniques and methods within my lab, as I say, to create capacity and expertise in conducting genomic experiments.
One of the genomics experiments which you have conducted resulted in a report titled Toxicological Evaluation of Emerging Chemicals in Municipal Wastewater Effluents Within the Georgia Basin, correct?
That's correct.
Mr. Lunn, if you could bring up tab 7 from our list, please. And so this is a report which did some work with effluent from the Anasis waste treatment plant and liquid waste from the Capital Regional District, correct?
That's correct.
I wonder if you could just, in fact, perhaps before we get into it, we could mark this as the next exhibit.
Exhibit 1046.
And the samples that were taken for this study were taken in 2004 to 2006, is that correct?
The samples were collected over, I believe, three sampling periods, 3 years, and there was a fall and roughly a summer campaign, if you want to call it that, where we looked at summer and winter variations in the effluent.
You could explain to the Commissioner briefly what the objectives of this research project were.
This project was funded under regionally Environment Canada regionally funded program, the Georgia Basin Action Plan, and again, through a competitive process, submitted in a proposal to use the methods that we've been working on, the genomic methods, to look and see if the techniques that we've been developing would lend themselves to looking at emerging chemicals of concern contained within municipal wastewater effluent. The study looked at non-lethal, started with non-lethal concentrations. So those concentrations, what we call the acronym is NOEC, no effect concentration. So fish were exposed to the highest no effect concentration, and concentrations that engineers at GVRD and now Metro provided us that were I guess relevant to receiving water concentrations downstream of the Anasis discharge. So using established testing platforms, we exposed rainbow trout to various concentrations of Anasis effluent for 8 days, and then over that 8-day period, fish were cropped or dissected before their livers were dissected and processed, and then a cohort of those same animals that had been exposed to the effluent were put into clean water, a depuration phase, if you want to call it that, for a further 8-day period. And the same thing was done. We excised out their livers from the various concentrations and then performed genomic analysis on it. The liver is a principal tissue that biologists and toxicologists look at because largely any contaminants or get metabolized and eventually end up in the liver. So the liver becomes a principal target tissue to look at potential endocrine effects or other effects as a result of exposure to low-level chemicals. How we determine the impact of the chemicals on the fish at the various concentrations is what we use what they call a gene chip or a gene array. And on that array, we had at that time we were doing the study, we had what we call a customized array. We had gone after specific gene targets or specific families of genes, whether they were stressor responsive genes, which are called heat shock proteins. We went after genes that were responsible for reproduction, vitellogenin, and other types of general genes, family groups that the literature in with which we had a fairly good understanding of how biochemically they responded to effects. So the cutting to the end point of this, at various concentrations, we saw different gene groups showing what they called it up-regulation or down-regulation. And all these expression levels are compared back to a control cohort that had the same testing conditions as with which the exposure concentrations, but obviously they just weren't exposed to any of the potential toxicants. So all the expression levels we saw are normalized back to the control group so that anything that we reported was relative or as a consequence of exposure to the various toxicants. So over the 16-day period, we saw certain gene groups come up, but for the most part, they returned back to background levels after when compared to the depuration phase. But what we did see was that there was spikes in immunoresponsive genes, which would be made good sense because, as I say, the fish were in concentrations of municipal wastewater level that would have triggered immunoresponsive processes to combat any potential infections and things. But the bottom line is that after the exposures, the majority of the levels of the gene expression returned to background levels, but there was certain levels of the vitellogenin proteins that did stay up. And the interesting thing about vitellogenin, it's a precursor to egg production in females. And the fish that we use were rainbow trout, and they're sexually immature and they're juvenile. So you should not be seeing vitellogenin showing up in two cases: A, at sexually immature fish, and also the fact that it was pretty ubiquitous across all the fish that we looked at. So it was interesting from that perspective that we saw the expression profiles that we did. But trying to relate that to receiving water impacts, a straight line correlation could not be drawn from our report.
Okay, thank you for that helpful explanation. Is understanding the potential impacts, sublethal impacts or cumulative impacts of certain chemicals, perhaps especially some of the emerging chemicals in municipal wastewater on fish, is this research, the continuation of research like this, important to that, furthering that understanding?
In my opinion, I think it's critical because historically the tests that we use in a regulatory arena and those used by proponents for complying with a regulation, the standard test is the acute lethality test. And majority of industries will pass that test because there is such strong enforcement on that. So they will not fail the traditional 96-hour LC50 test, which is a test of lethality. But we know that certainly within the complex mixtures of various industrial compounds, particularly municipal wastewater, there's emerging chemicals of concern in ultra low levels that have been demonstrated either individually and individual scientists' research that are indicative and conclusively causing endocrine disruptor effects. But also the fact that the ability of a lot of these treatment systems do not or cannot remove or treat a lot of these emerging chemicals of concern. As I mentioned, pharmaceuticals are just one classification. There's another group of chemicals that are currently being used in commerce of a thing called nanomaterials or nanoceuticals that the pharmaceutical industry is using nanotechnology to further increase the efficacy of delivery of their target compounds to for drug treatments. But again, they're excreted out into municipal wastewater and discharged into the effluents, and that the conventional fish bioassays will not be, and the endpoints associated with those bioassays will not be able to detect those.
Okay. Let me ask you this in the context. Can you relate the degree of funding that's provided to you with your ability to continue or conduct further research of the type that was conducted in this study?
With respect to genomics, the STAGE program, as I previously mentioned, is the only source of genomic funding within Environment Canada, and that at the end of this fiscal year is coming to a close, and a new program is thought to be on coming forward, but it will be with the five science-based departments of Environment Canada. So and the emphasis is not on aquatic environmental toxicology.
Dr. Ross, I wanted to ask you about a study or a piece of research you did, Exhibit 833, please, Mr. Lynn. In 2002, Dr. Ross, you did some research with respect to late-run sockeye and produced a paper with others titled Late-Run Sockeye at Risk, an Overview of Environmental Contaminants in the Fraser River Salmon Habitat.
That's correct.
That's the paper we see on the screen there?
Yes.
Okay. And this was, as opposed to primary research, really a literature-based risk assessment, correct?
Yes, that's correct. We felt a little bit blind and in the dark with regard to what was happening with the late-run sockeye crisis that started in the late 1990s and was leading to the loss of tens of millions of fish over a number of years. And it was an altered behavioral or return migration phenomena that was taking place, and it was the subject of considerable concern, of course, to the Department of Fisheries and Oceans, Pacific Salmon Commission, and other parties. And we thought it would behoove us to at least get a cursory risk assessment conducted to try to give a little bit of guidance on possible chemicals of concern in the Fraser Valley that might be leading to this effect. We have 23,000 chemicals on the domestic substances list in Canada. Many of those chemicals are either used or end up in Fraser River salmon habitat. And to understand and or prioritize which of those chemicals might present a risk or a significant risk it was something that we felt we could at least try to do with this kind of approach.
Now, we'll get into the specific chemicals in a second, but just so the Commission understands, a number of the chemicals that attracted your attention in the study, one of the sources of them is municipal waste. Is that correct?
That would be correct.
Okay. Within the body of this study, and specifically in the abstract, you summarize your conclusions and your recommendations for future research, correct?
Yes.
And you recommend that if we're looking at wastewater research in a couple of areas, the first was a need for further research of the impact of exposure levels for certain key chemicals, including persistent organic pollutants and PBDEs. Is that correct?
Yes.
You recommended further research with respect to surfactants.
Yes.
And you also recommended further research with respect to certain pharmaceuticals and personal care products, correct?
Yes.
And are those all chemicals which can be found or sourced in municipal wastewater?
With little doubt, that would be the case.
And have the recommendations which you made in your 2002 paper been followed up on by the Department of Fisheries and Oceans?
Well, I would say that some of the recommendations have taken the form of specific research projects undertaken by myself and collaborators immediately. Some related to current use pesticides in salmon. I would say yes on that front. There's a very small funding envelope within DFO nationally, $300,000 a year for providing information on biological impacts of currently used pesticides in support of the Pest Management Regulatory Agency. Some other research projects, either led by myself and or by my colleagues at the Institute of Ocean Science, where there are 2 or 3 other contaminant specialists, Dr. Economou, who's done some work on pharmaceuticals, Dr. McDonald and Dr. Johannesson, who do some work on transport and fate of certain chemicals in the environment, notably PCBs and PBDEs. These 3 colleagues of mine do not look at the effects of those chemicals, but they look at where they end up and why they end up there, and environmental processes that might shape their fate in the environment. So I think it has, some of the recommendations have borne fruit, if you will, but that there has been no impacts on envelopes, funding envelopes, grants, or priorities to direct any of this research.
I'm going to move away from your research now and ask you some questions specifically about the impacts of municipal wastewater on Fraser sockeye. Perhaps since you've got the microphone on, Dr. Ross, I'll start with you. Based on your experience and your research, are you able to offer an opinion as to whether municipal wastewater has harmful effects on Fraser sockeye?
Well, as we have discussed already today, we don't really have a mandate to look at end-of-pipe discharges and effects on salmon. But certainly, if we look at some of the chemicals of concern in the wastewater stream, there are a number of classes of concern. And I think the way I would capture it is in the absence of much direct evidence from the Fraser River system, we have to rely on some of the literature from other parts of the world. And then we have to do a sort of a risk oriented approach to trying to rank which types of chemicals might present the greatest risk here. I should point out that there have been several examples from other parts of the world that would underscore the potentially important threat that wastewater treatment streams do present to the health of fish. The widespread feminization of fish has been taking place in the United Kingdom, and this is accentuated downstream of municipal wastewater treatment plants. And this has been surmised to be largely due to the estrogenic nature of the wastewater stream. Estrogenic nature simply means the stream has estrogenic potential and can feminize male fish or alter reproductive health in both male and female fish. That estrogenic nature will come from natural estrogens from human wastes, from agricultural animals, from birth control pills, but also a lot of pharmaceuticals, synthetic musks, and a lot of the persistent bioaccumulative chemicals as well. So there are a lot of chemicals of potential concern, I would say, in the Fraser River system, and certainly being released from wastewater treatment plants.
Okay, thank you. Dr. Ashley, I'm going to ask you to weigh in on this question, and if you want to sort of address it in two phases, both acute and chronic impacts, I'm content for you to do that as well, because I understand your thinking might go along those lines.
The question being relevant to Fraser River sockeye.
Yeah, municipal wastewater harmful effects, if you believe there are any on Fraser sockeye.
Potentially there's an acute toxicity issue with some of the high concentrations of ammonia that are discharged from a couple of the wastewater treatment plants. One large one at Anasis, which discharges about 500 megaliters per day, and then the Lulu Island treatment plant. They're known for having high ammonia discharge concentrations. Those could potentially cause acute toxicity issues, and the other ones, as Dr. Ross had just said, is concern about emerging endocrine disruptors because of this widespread knowledge around the world, in the UK and in Australia, of sex reversal.
Mr. Van Agelen, do you have anything to add to those two answers?
The only thing I would add to that is that one of the things that researchers have discovered is that it's the life stage is at which the organisms are exposed is critical. So the developing fish or early life stages are much more vulnerable, or thought to be more vulnerable to low-level exposures of pharmaceutical personal care products, or collectively xenoestrogens, as Dr. Ross mentioned.
Okay. I'm going to come back to the ammonia issue in a second, Dr. Ashley, but before we come to that issue, on the general issue of the harmful effects of wastewater, to the extent there are any on Fraser sockeye, does the state of research impact on the certainty with which you can offer an opinion in this area?
I think there's a fair amount of published literature that use a weight of evidence approach to say if there is an effect or not. And so the answer is yes.
Okay. Dr. Ross?
Well, again, I speak with some ambivalence just because of the lack of a tremendous amount of research in this field in the Fraser Valley. But if I imagined, as my colleague Mr. Van Agelen here had inferred, developing eggs and fry in the headwaters down through smoltification at a very small size, young age, within a year, in through the Fraser River and estuary and out into the open ocean. As I understand it, there are 90 wastewater treatment plants in the Fraser River Valley. And so if we're talking about point source discharges and certainly the high-level public concern about acute toxicity related to a single point source, I think I would step back a little bit and raise some concern about 90 point sources, of which certainly the lower Fraser estuary has the largest by volume discharge. But I would point to the cumulative exposure from a very young age to some of these chemicals of concern throughout their early life and certainly upon return. And some of the chemicals we're talking about we might categorize as persistent. That is, they don't break down. Once they're released into the Fraser or into the Strait of Georgia, they're around for decades, if not centuries. These would include the dioxins, the PCBs, the organochlorine pesticides, the PBDE flame retardants. On the other side of the spectrum, we have chemicals that might simplistically be viewed as non-persistent or less persistent. But when I look at 90-point source discharges for wastewater treatment plants, those non-persistent chemicals become pseudo-persistent because they're chronically being released, and they're being released at virtually every point along the freshwater habitat for these migrating sockeye.
Thank you. Mr. Van Agelen, do you have anything to add to?
I basically just echo with what Dr. Ross said, is that, it's the persistent, low-level, continuous exposure of a toxicant that can result in certainly from molecular or sublethal effects that could be seen. As I say, that would be my take, is that the sum total of all the discharges in coming through with low levels of various contaminants could contribute to the molecular or sublethal or chronic effects that maybe we're seeing.
Dr. Ashley has identified ammonia as one contaminant of particular concern. Do either of the other two on the panel have any particular contaminants or chemicals that they have identified that are of particular concern and think warrant special attention?
I'll speak up. As I say, I think that municipal wastewater is, there's all kinds of things you could say for it, but essentially it's a cocktail. It's a cocktail of everything, whether it's particularly as with respect to drugs that people take, to things people pour down the drain. I think that most people view sewage treatment plants as this magical place where everything gets cleaned, and essentially the treatment of sewage really hasn't changed all that much since I think the Romans first introduced us to biological treatment of human waste. So essentially, we can clean up the conventional things like suspended solids, biological oxygen demand, and to a certain degree control for ammonia. And those, as I think Dr. Ashley mentioned, are all acutely toxic parameters. And those plants that are efficiently run or that have upgraded to treat for those, what I would call conventional parameters of toxicity and effluent quality, are in check. But it's, as I say, the myriad of chemicals that society puts into a treatment system is, you could spend all day listing them and you still wouldn't come up with the sum total of everything that's in a complex mixture.
Okay, thank you. Dr. Ashley, you've identified the Annis's treatment plant and the ammonia issue, which we'll come to in a second. Dr. Ross or Mr. Van Agelen, are there particular waste treatment facilities or discharge sites that you think warrant particular attention or are of particular concern to you, thinking from a Fraser sockeye perspective?
Well, this is a little bit outside my line of expertise, but I'd be looking for those discharging the most in terms of volume, and then I'd be looking at treatment level. That'd be two features. So Annis's Island certainly comes to mind in that respect. I'd also be looking at the vulnerability of the receiving environment, because oftentimes when one looks at sort of national minimum standards or national rules of engagement, one's looking at a certain common denominator. And unfortunately, a lot of these plants, well, none of these plants discharge into the same body of water. There's different dilution levels, there's different flow rates, there's different depths, and different species inhabiting those environments. So those are some features I'd be looking for with regard to sort of a hazard-oriented approach to identifying wastewater treatment discharge points of concern.
And using the criteria you've expressed, are there any particular plants that you identify as ones that perhaps would raise red flags?
Well, the overwhelming number amongst those 90 wastewater treatment plants in the Fraser Valley are secondary or even tertiary. And secondary or tertiary it doesn't solve all the problems, but it does reduce the inputs of a lot of harmful compounds, many of the ones we're talking about today, with some variation. And we can certainly speak to that a little bit. But when we get down into the Fraser Estuary, where we have primary treatment, obviously there is less retention of some of the chemicals of concern to sockeye salmon.
Okay, thank you. I want to come back to the ammonia issue now, Dr. Ashley, and wonder if you can just explain to the Commissioner what your concern is with respect to the ammonia and the waste treatment plants you identified.
It's, as discussed, it's one of these older contaminants that have been known for a long time, and it's acutely lethal depending on a combination of pH and temperature. And the design of the plants at Anasis and Little Island are not particularly effective at converting it to a non-toxic effect. And so they discharge at fairly high concentrations in the effluent stream, 25, 30 milligrams per liter. And then the assumption is that will be adequately diluted to the point where it's non-acutely toxic to salmonids. Now that's based on an average, and remember the Fraser River flows downhill and then it flows back in, and then there's a slack tide period. And the concern is that I've been out during the winter time when the Fraser's at its minimum flow, down around 1,000 cubic meters per second at a slack tide. Basically there's a couple-hour period where the effluent is just pouring out and it's not moving one way or the other. And so that the concept of a dilution zone sort of disappears temporarily, and whether that would be a stressor on salmonids that were resident at that time. Obviously, migrating sockeye are not there at that time. This is more of a resident fish species in the area.
Okay. From a sockeye perspective, are you aware of any evidence that links harmful effects from ammonia from those two plants to Fraser sockeye?
No.
Okay. Is this something you've sort of surmised from looking at the evidence of what's discharged and knowing what you know about Fraser sockeye?
It's not specific to sockeye. It's just the fact that the concentrations are so high that the plant is not particularly well designed to remove ammonia, and that Metro had to develop a separate acute lethal toxicity test to deal with ammonia because the standard Environment Canada test was failing the LC50 test.
Okay. You addressed some of your concerns with respect to ammonia in the Anasis plant at the Speaking for the Salmon conference, correct?
That's correct.
And Mr. Commissioner, just for your note, Exhibit 12 contains that summary of the presentations from that conference. Now, Anasis Island, you've talked about the different technologies, and just so we're clear, when you refer to the different technologies, you're referring to the fact that Anasis has a trickling filter design as opposed to an activated sludge design?
That's correct.
Okay. And is the retention period shorter with the trickling filter design?
Yes, there's two types of retention coefficients used. One is the hydraulic retention time, and one is the solids retention time, and both are very short in a trickling filter solids contact design, such as Anasis is, relative to an activated sludge design.
Thank you. The city test conducted on the ammonia, or conducted with respect to ammonia for Anasis, or to the discharge with respect to Anasis. You know something about that testing change?
I wouldn't say change is an accurate word. There was what they call a pH control method that was developed actually out of my lab to look at those effluents that where I have to back up, Your Honor, that in a conventional bioassay for doing compliance monitoring, the test solutions are aerated at a prescribed rate of 7.5 mils per minute per liter for the duration of the bioassay. And ammonia, as Dr. Ashley mentioned, is a very complex, chemically complex toxicant, and that can, depending on the state of pH, the toxicity can be hugely more toxic as pH shifts. And what we were seeing as a result of the bioassay is that the aeration rate was driving off or gassing off. I hate to get into scientific jargon, but the carbonic acid ions in the water solutions was being driven off and causing a shift in the pH in the solution, and it was converting the total ammonia contained in the solutions to a much more toxic state, which is what they call the unionized ammonia concentration. And this was causing, in some instances, erroneous results. In other words, samples were being shown to be more toxic than with which they were. So we developed a method to control the pH drift in the bioassays by injecting carbon dioxide to compensate for the carbonic acid that was being driven off. So basically, we're hence pH control. So with this method, we were able to determine if there were acute toxic concentrations of ammonia in the sample that that weren't being converted as a result of a pH shift. So essentially, a method to ensure that that the method did not report erroneous results, and that if there was entrained toxic levels of ammonia or unionized ammonia, that we would pick it up in our compliance test.
And that test, both originally and as modified, is designed to test for acute toxic effects, lethality.
That's correct.
I'm going to ask that you bring up Exhibit 616A, please, Mr. Lund. Dr. Ross, I'm going to ask you some questions about this document. It's a memorandum to the Minister dated December 3rd, 2009 from the Department of Fisheries and Oceans. Are you familiar with this document?
Only through having seen it as an exhibit.
Okay, and you've now reviewed it?
Yes.
Okay. You've given us your views on the potential connection between municipal wastewater and Fraser sockeye. This is a memorandum addressed to the Minister, titled Factors Affecting the 2009 Fraser Sockeye Return. I wonder if we can flip to the next page, please, and zoom in on the analysis DFO comment and then number 1. That's sufficient as it is. Okay, the first point under this heading reads, the following factors are unlikely to have contributed to the poor 2009 return. Point 1, pollution in the Fraser River. Do you support this statement as written in this memorandum?
Well, I guess I'd have to say that I haven't seen any data that would empower me to suggest that there's no evidence for pollution having played a role.
Okay. Did you participate in the September 2009 DFO Science Workshop?
Yes, I did.
And you made a presentation there?
Yes, I did.
And was your presentation consistent with the message as articulated in this memorandum?
I can't say that it is completely consistent. I think my sense from this reading this statement, pollution of the Fraser River, there is no record of any Fraser Basin-wide environmental incident that could have impacted fish. It strikes me that the author or authors are referring to an absence of any reported spills or any broad-scale impact. And the fact that with the sockeye returns in 2009 being so poor, the author or authors contributing to this were probably looking at a fairly simple A plus B equals C relationship. That said, I remain fundamentally concerned that environmental concentrations of a number of endocrine disrupting compounds, both persistent and non-persistent, can adversely affect the health of sockeye at different life history stages and may increase the risk of mortality through a number of developmental means. The absence of a spill or the absence of a fish kill, to me, does not indicate that pollution can play no role in affecting the lifelong health of a fish.
Could we have our list of documents number 6, please? Dr. Ross, you're familiar with the Canadian Council of the Ministers of the Environment and the work that they did to come up with a Canada-wide strategy?
Yes.
And that's a collection of all the ministers of the environment from each of the provinces and Canada, and they got together and worked out a strategy to approach wastewater treatment in Canada, correct?
Correct.
And speaking very generally, it was designed to create consistent, nationally applied criteria and rules for the treatment of wastewater.
Well, as I understand it, that is essentially the hallmark of this CCME effort, a national minimum standard to make things easy in terms of design, operation, monitoring, and assessment.
Okay. And what flowed out of that Canada-wide strategy was the drafting of what are now proposed wastewater system effluent regulations, correct?
Correct.
And those are somewhat analogous, although certainly not identical to the regulations that were crafted for the pulp and paper industry and metal mining, correct?
Correct.
We have the draft regulations or the proposed regulations, Mr. Commissioner. I'm going to suggest those become the next exhibit.
1047.
Dr. Ross, you had the opportunity, along with some of your colleagues, to review these draft regulations and offer your comments on them.
Yes, we had a very short window to respond to the proposed wastewater systems effluent regulations.
And you and some of your colleagues reduced your comments to writing?
That's correct.
And those were put together in a memorandum addressed to Robin Brown?
That's correct.
If we could have our list of documents number 30, please. And this is a copy of that memorandum?
Yes, it is.
Okay, if that could be the next exhibit, please, Mr. Commissioner.
So 1048.
I wonder if you'd just take a moment now, Dr. Ross, and address the Commissioner on any comments you have on the proposed regulations and any concerns you have about their adequacy, bearing in mind that our focus is Fraser sockeye.
Well, I should start by stating that I played no part in any of the CCME discussions or deliberations leading up to the Canada-wide strategy or the proposed wastewater systems effluent regulations that are not yet in effect, but will take place under the guise of the Fisheries Act. So my only involvement on this file would be this rather rapid turnaround review of the proposed regulations. And as scientists do in terms of peer evaluation, we look for gaps, data gaps, and the like. I think my take-home message was, well, it's important to have a clear set of terms of engagement, and a national strategy and national regulations would help on that front. I did express the concern that a national minimum standard does not necessarily upgrade the performance of all wastewater treatment plants. Certainly a minimum standard of secondary will reduce the risk to some aspects of Fraser River sockeye habitat because an upgrade from primary to secondary will reduce the release of a number of contaminants of concern. Specifically, the proposed regulations cover the kinds of chemical constituents or activities thereof that we've been worried about for dozens, if not hundreds of years by default. That's suspended solids, total residual chlorine, deionized ammonia, and biological oxygen demand. Sorry. But they do not, in looking at these four primary conventional pollutants, there is only fleeting mention of site-specific impacts and concerns, only fleeting mention of emerging chemicals of concern, such as the flame retardants or the pharmaceuticals. The reporting of monitoring data appears fairly elementary, and the effects monitoring ceases to be a requirement if there are no adverse impacts observed after a certain number of years. So I did have some concerns, and in looking at government operations and the way in which we do science to look at the broader risk to the receiving environment associated with contaminants, I think I harken back to the question of cumulative impacts. These regulations were not designed to protect salmon. They were not designed to prevent cumulative impacts associated with multiple treatment plants. and they were not really designed to deal with the concerns that I have about bioaccumulation and biomagnification in food webs. A lot of chemicals, even present at very, very low concentrations, if they're very persistent, they can get into food webs and be found at very, very high concentrations in some organisms of concern, such as marine mammals. So we did have a number of concerns about a national minimum standard, or this common denominator, and we did have some concerns about the fact that site-specific impacts would be subject in some form to local regulatory frameworks, but that remained to me a little bit unclear. There may be additional ancillary discussions going on to render this a little bit more seamless with regards to provincial regulations and legislation, or site-specific requirements under a liquid waste management plan for certain waste treatment plants.
There's just one other issue I wanted to have your comment on, and that is the issue of biosolids and the extent to which they're dealt with by the regulations. Do you have a comment on that?
Well, as I understand it, there is no mention of what happens with the retained sludge, and I do have a concern of, if we're talking about persistent contaminants, the PCBs, the dioxins, the furans, the organochlorine pesticides, the perfluorinated compounds, the polybrominated diphenyl ethers, those are flame retardants that are only recently under the regulatory microscope in North America. These are all very, very persistent compounds. Upgrading blindly from primary to secondary to tertiary does not degrade these compounds, does not break down these compounds, but it does retain many of these compounds because these chemicals are all persistent and they bind to organic materials or to fats in the food web. The fact that they're so persistent means there's only one way to get rid of them, and that's with incineration at 1,000 degrees Celsius or higher. The half-life of most of these chemicals in the environment is in the order of hundreds of years. If sludge is being retained, biosol is being retained, and transferred to agricultural lands, forestry lands, mine reclamation projects, or landfills, those chemicals are maybe not coming out the pipe anymore, but they are entering the environment. They're simply being cycled to another part of what is likely to be Fraser River sockeye habitat.
Okay, thank you. Dr. Ashley, you've heard Dr. Ross's comments on the proposed regulations. I wonder if you have any observations to add.
I agree with everything he said. I think obviously the country needs a national standard because there's many parts of Canada that have either no wastewater treatment, like Victoria, for example, or places in the north that have fairly rudimentary. But to say that a movement to secondary is going to solve all the concerns about potential contamination in the environment is not true.
Does that conclude your comments?
We'll probably get back to this one more later.
Yes. Yeah, we will come back to the different treatment levels, and I'll allow you to offer your opinion on what's advisable in that regard. Mr. Van Agelen, do you have anything to add with respect to the proposed regulations?
No, I concur with what Dr. Ross mentioned, and just with the added bit that our understanding of how to treat the chemicals is not well understood, and as Dr. Ashley mentioned, blindly saying or going to secondary or tertiary treatment is not the cure. It's better, but it's not for the 21st century knowledge of chemicals as we know it now. Secondary and tertiary is not going to take care of our problems, and as we were talking about biosolids, yeah, I have grave concerns about how we are regulating or administering biosolids as a soil additive, and as Dr. Ross mentioned, I think it's just a means of transferring the potential toxicants from one source to another, and subject to rain events and other erosional events, it could liberate and start the migration of low-level contaminants that have been bound up in the biosolids, and science has proven that there's certainly some of these emerging chemicals of concern have a high binding affinity to the particulate matter in treatment processes, and that is all part and parcel of biosolids.
Thank you. Dr. Ashley, you have an engineering background, so I'm going to address the next questions primarily to you. You mentioned Capital Regional District or Victoria, which has very limited waste treatment, not even to the primary level as I understand it. We have Iona, which has primary, and then there's some different secondary options available, activated sludge and trickling filter. I'm wondering if you can walk the commissioner through sort of the upgrade from nothing to primary and primary to secondary and secondary to tertiary, and explain to him what is gained in terms of removal of contaminants or matters of concern to sockeye with each of the upgrades.
All right, we'll use the conventional terminology as this primary, secondary, tertiary, but I'd like to mention that there's within the engineering circles, there's different ways of looking at that, and I'll bring that in at the close of this section. Capital Regional District basically just has a screen mesh, and so that's called preliminary screening. So basically it's to catch rags, sticks, things like that are fairly large scale, and that's basically it. So that's preliminary treatment. Those exist in all wastewater treatment plants, just as a matter of protecting pumps and equipment downstream within the plant, so they don't get jammed up, and the rest of the effluents just sail right through. So it's basically a coarse screen. Primary treatment is basically designed to retain the effluent for a certain period of time and settle out the suspended solids, and so it has a settling rate of around 60 or 70% of the suspended solids settle out, and around 30 or 40% of the biochemical oxygen demand. That's carbonaceous material in the wastewater that consumes oxygen in the receiving environment. The movement to secondary, the containment is held in a plant longer, and so the suspended solids removal percentage goes up to around 90%. The removal of BOD5 goes up to 60, 70, even 80% in good plants. But basically, all of the all the remaining pollutants continue to sail right through and be discharged. If one moves to tertiary, the conventional sort of designation for tertiary is that it removed nitrogen and phosphorus, and so that was a tertiary plant. So basically those were a secondary plant with an add-on for nutrient removal. And next step beyond that, not seen very often in Canada, is called a quaternary treatment plant, where it would actually remove some of these chemicals of concern. That's used in places like Israel, in Southern California, San Diego, where they're water short. The term that's emerging now is within the engineering circles is often to refer to conventional wastewater treatment, because secondary treatment is so normal these days in many parts of the world. I mean, it was discovered in I think 1910 was the first activated sludge plant. It's referred to the term advanced wastewater treatment and use the best available technology for the day. And so those two terms can be used interchangeably, but that's basically the difference between preliminary, primary, secondary, tertiary, quaternary, and then the sort of discussion to move to advanced wastewater treatment plant using all of the technology that's available today and proven.
Okay. And in terms of the cost differential, what is the cost differential between sort of secondary or moving to tertiary, or from secondary to best available technology?
It depends on how the plant is designed. Some plants are very adaptable towards upgrade to tertiary. Conventional secondary treatment activated sludge plants, there's even a company that specializes in rearranging the flow pattern within the plant with very little new tankage that essentially converts them to biological nutrient removal designs. Quaternary is the really advanced wastewater treatment plants. Those are usually add-ons as the effluent has to be fairly clear before they can be used for absorption of some of these chemicals of concern at the end. Activated carbon charcoal columns can be used. There's other suggestions that ozone, because of its strong oxidizing capability, can be used once the effluent has passed beyond the tertiary stage. Other design plants of secondary are relatively expensive to upgrade to nutrient removal, just because of the inherent design. So we're referring to trickling filter plant designs are not very easily converted to nutrient removal because of their inherent difference in design between a trickling filter and an activated sludge. So you might be looking in some cases at a 10 to 20% cost increase to go from secondary to tertiary for an activated sludge plant, whereas a trickling filter secondary plant, it might be half the cost of the plant again to convert it to nutrient removal and more advanced containment for emerging chemicals are concerned.
If we're looking at a plant that's going to be starting from scratch, as I think we'll hear Iona will likely be, what sort of cost differential would those footing the bill for that be looking at if they were to go beyond secondary to best available technology?
Designed at scratch is the time to make the decision, because that's when the cost differential is at its very least, and so it would be probably in about the 20% range to go to advanced plants over a conventional secondary. It would be very foolish to just build a secondary treatment plant, because by the time it was built, it would be technologically out of date given the new regulations that will be coming down in terms of capture of emerging chemicals concern.
Okay. Speaking of the Iona plant and the upgrade that is contemplated for that, from a Fraser Sockeye perspective, how important is it to look at something beyond secondary for that plant? I'll ask you first, Dr. Ashley.
Because it discharges into the estuarine marine environment, it's of less immediate concern than something that was discharging directly into the Fraser, but it adds to the burden of persistent chemicals in Georgia Strait, which obviously would have a concern with the life history of sockeye.
Dr. Ross, do you have anything to add to that?
Well, for Fraser River sockeye, of course, Strait of Georgia is part of their habitat, and releasing compounds that are ending up in the Strait of Georgia may be out of sight, but it's not entirely out of mind. I can point to our recent study that's in press right now that describes PCBs that have been banned since 1977 and PBDEs that are only now becoming regulated in Canada and have no regulations in the United States. These are both very persistent bioaccumulative and toxic compounds of concern to the food web, because they get into the fats at the bottom of the food web. They biomagnify. PCBs have heavily contaminated southern resident killer whales because of the legacy of the use of that chemical. PBDEs have been doubling every 3.5 years in harbor seals in the Strait of Georgia, and are now probably plateauing or dropping. Our study examining PCBs and PBDs in sediments in the Strait of Georgia show very high concentrations around the eastern shoreline of the Strait of Georgia, notably around the outfalls and into Burrard Inlet. So very, very high concentrations of PBDs, much higher than we would expect based on our observations with PCBs, which indicates to me very strong localized point sources of flame retardant chemicals that are coming out of day-to-day use, computers, furniture, carpeting, textiles, electronics, automobiles, landfill leachate, clothing even, and these things would get into the wastewater stream. Whether it's primary or secondary, there's certainly a large fraction ending up going into the plume, into the stream, and into the Strait of Georgia. Concern about that is that over time we're building up a reservoir in the Strait of Georgia, and over time that will start to present biological risks to the critters that live and or transit the Strait of Georgia.
Okay. If we were looking at priorities, where would you rank the priority in your priorities, the need to upgrade Iona as compared to perhaps other concerns that might exist with facilities, wastewater facilities?
Well, other concerns to me would extend far beyond the wastewater treatment
Yes.
plants. Of course, pulp mills are huge producers of some of these estrogenic compounds of concern. Some of the pesticides are used in agriculture and forestry are of concern. But in terms of wastewater treatment plant, I would say that anything that reduces the release of compounds of concern and upgrades from primary, secondary, and tertiary would all reduce the inputs of current use pesticides, of persistent compounds, of pharmaceuticals, etc., but it doesn't necessarily solve the problem. I think I would point to a couple of other activities that there are times when municipalities or regional governments get sort of blamed for these chemicals, but the fact of the matter is Metro Vancouver or Capital Regional District did not produce these chemicals of concern. They're stewards of our wastes, and as such, there's a heavy responsibility in terms of trying to have wastewater treatment practices that eliminate potentially 10 or 15,000 chemicals of potential concern in terms of sockeye. So I think I would point to CEPA, the Canadian Environmental Protection Act, which has as part of its direction chemical regulation. And PBDEs are a good case in point. PBDEs are starting to be regulated in terms of CEPA. So there is a chemical regulation side of things to prevent chemicals from getting into the wastewater stream at the beginning of the day. And the second thing I would point to would be source control. I know that Metro Vancouver and Capital Regional District have very strong, very important source control programs. And source control can target photo finishing labs, can target dentists, can target automobile repair shops, etc., and try to prevent some of these chemicals from getting into the stream. It's easier to process them and reduce risk at that level. So I may be wandering a little bit for your purposes, but those are a couple of thoughts I did want to get in there.
No, it's helpful, and it saves me a couple of questions, which I was going to come to. Mr. Commissioner, it might be an appropriate time for the break.
Hearing will now recess for 15 minutes.